there's about one thrombocyte for every 10 or 20 red blood cells. So yeah, maybe 5 to 10% as many of these as there are red blood cells. So they're a little scarcer. But thrombocytes, also known as platelets, really only have one job. They're involved in blood clotting. These aren't really true cells at all. These are really fragments of a larger cell called a megakaryocyte. Megakaryocytes split off platelets and shed them into the blood. So they have no nucleus, they have no organelles, they really have no cellular structures at all. They're just pieces of cells. Mostly what they are is full of granules of various sorts of chemicals that will be involved in blood clotting. They don't last very long. Within the body, they last about 9 days. And in general, your body's highest goal is to have them go those 9 days without doing anything. When you activate platelets, it's because something is wrong. Because a vessel has sprung a leak somewhere. And platelets are part of the process by which you avoid losing all of your blood through blood vessel leaks. The process by which they do that is pretty interesting. Generally, it's usually called the coagulation cascade. And we're going to go over it in moderate detail. There are parts of it that definitely fall into that area that I call Shazam type mechanisms, in that they are complicated and we actually know pretty well how they work, but they are way more complicated than we need to get into here. So let's sketch out a blood vessel. So these very thin cells I've drawn here are meant to be the endothelium, the single cell layer that lines the inside of a blood vessel. Now, what's really important to note is that in every blood vessel, the endothelium, just outside the endothelium, is a layer of fibers called the basement membrane, or the basal lamina. That's mostly made of collagen, often with some elastin in it. So we'll draw that in green here, if the screen marker works, which it so far is not. So the basal lamina is largely composed of collagen, which is a protein. And it's maintained by cells called fibroblasts. Sketch in a few fibroblasts. That one's better. These are the cells that make the basal lamina and repair it. Now, it is important that this collagen is normally sealed away from the blood by that endothelium. So normally, as platelets are circulating here, these endothelial cells secrete chemicals called prostacyclins, which keep the platelets quiet. They're very soothing to platelets. Platelets don't do anything as long as the prostacyclins are there. However, if we end up with damage to the vessel wall, there's going to be, as far as I can tell, 3 different sets of reactions that are all going to happen and all going to interact that in the end will result in the formation of a blood clot that will stop the blood from leaking through here. So, process number 1. When platelets run into collagen, platelets have receptors for collagen, and when they run into collagen, they become activated. Several things happen. Number 1, they stick to the collagen.
And this is coagulation cascade.
Yeah, this is one of the pathways involved in that process. Not only do they stick to the collagen, but they become sticky to other platelets. They start putting out proteins on their surface that other platelets will stick to and in turn activate themselves. And finally, they start secreting signal molecules. Which start interacting with various proteins in the plasma. So, area number 1 is that our platelets start sticking to that collagen and each other, forming what's called a platelet plug. The second part of the pathway is that the protein collagen also interacts directly with some of the proteins in the plasma. So here we have collagen interacting with the plasma proteins, starting a series of reactions there, sometimes called the intrinsic pathway. So far, platelets have stuck to the collagen and started gathering platelets. In addition, the platelets are releasing molecules into the plasma which start interacting with plasma proteins. Pathway number 2. The collagen itself interacts with some of the plasma proteins, starting other reactions. I'm being very vague here because this is the Shazam type mechanism. The plasma protein interactions here are insanely complicated. The third pathway is that a protein called tissue factor, which you find on fibroblasts, also interacts with plasma proteins. Sometimes called the extrinsic pathway. So 3 different things have started to interact with different proteins in the plasma. And we start this really complicated dance known as the coagulation cascade. Now, I need to add a few more things in here. The activated platelets start to gather a protein called fibrinogen, which is dissolved in the plasma.
Is this a separate step or?
It's part of that first, the platelet aggregation process. So fibrinogen is shown there in red. Fibrinogen starts to come from the plasma and stick to the platelets. Good so far? Now, these 3 different pathways all start to interact with each other. Also involved vitamin K and calcium. They all go into this ridiculously complicated set of interactions that ends up finally producing a protein called thrombin. So the situation so far, we have our squishy platelet plug made of all these little things that are sort of blocking the tear. The fibroblasts have activated certain plasma proteins, the collagens have activated other ones, the activated platelets are sending out signals. All of this causes interactions with proteins that are in the plasma already, mostly from the liver, and end up producing a protein called thrombin. Thrombin is an enzyme, and it turns fibrinogen into fibrin. Fibrin forms long fibers which are tough and not soluble. They lace together through the platelet plug. They start catching red blood cells, and they form a big mess we call a blood clot.
So you said thrombin is an enzyme which turns what?
It turns the fibrinogen that the platelets had gathered into fibrin.
Okay.
Notice the name, fibrinogen, fibrin generator. Fibrin strengthens the platelet plug, starts catching erythrocytes, and we get a blood clot. That blood clot can grow pretty big. It can block a whole vessel. I've seen a couple of unpleasant videos on the internet where people are pulling blood clots out of people's veins and they can be that long. So yeah, they're kind of icky. But they're useful for stopping leaks. They're bad when they kill you, which they can also do, because they block the flow of blood quite effectively. Now one of the last things, part of the neat things, is that when the fibrin forms, and we form the clot also traps a protein in the blood called plasminogen. So this blood protein called plasminogen gets trapped in the blood clot. Later, when the blood vessel has healed, when the endothelial cells have regrown and we've sealed up the breach, plasminogen gets turned into plasmin, which dissolves fibrin. So the blood clot traps the inactive form of the enzyme plasmin. Once the vessel is healed, the plasminogen activates the plasmin, which breaks up the fibrin and releases the clot, and it breaks up and drifts away. Yeah?
Where does the plasminogen come from?
The liver, as far as I know. It's circulating in the blood like the fibrin is. In that form, they don't do anything. If you've heard of somebody who's a hemophiliac, that's somebody whose blood doesn't clot properly. Hemophiliacs are usually missing one or more of the various plasma proteins. They have a mutation which causes one of those, what they call clotting factors, to either be absent or less functional. Depending on the mutation, that can mean their blood doesn't clot at all or doesn't clot well. If your blood doesn't clot at all, that is a serious problem. You can bleed to death from a minor cut. So people who have severe hemophilia have to be very, very careful. It's also one of the main targets for gene therapy. In theory, all you have to do is get the gene for the good version of the clotting factor into the cells and get them to incorporate it into their DNA, and you've cured the condition. Easier said than done. Last I heard, there had been at least one successful curing and one where they accidentally gave the person cancer.
Nice.
Yeah, so gene therapy is still kind of, a young process, but potentially, probably within your lifetime, it will be used to cure genetic diseases. There's no reason it can't work, it's just not quite there yet, but rapidly improving. So think about it, cystic fibrosis, curable. Hemophilia, curable. A lot of these relatively simple genetic diseases, fixable, would be nice. Okay. Anything else? Yes?
Would you say this whole process is like step 4, or is it part of like step 3?
It's what happens when steps, the 1, 2, and 3 pathways all come together. They converge on that common thing and form thrombin. Alrighty. Good enough for one day. Okay.